things.
Yeah.
Yeah.
Graduation requirements.
Stuff.
So yeah, thanks for coming everyone to the first seminar of 2025. And Karen, our student coach.
Cool. Well, hi, everyone. Thank you for coming again. Hope you had a great start to the new year. Today, we have the great pleasure of welcoming Dr. Angela Brooks from UC Santa Cruz. And Angela actually did her undergrad here at UCSD in bioinformatics, and she went on to pursue a PhD in molecular and cellular biology at UC Berkeley with Stephen Brenner. And after grad school, she headed to Boston to work with Matthew Mayerson at the Dana-Farber Cancer Institute, where she studied the cancerous effects of mutations in U2AF1 and SF3B1. And Dr. Brooks then returned to California to join the faculty at UC Santa Cruz, where she is a professor in biomolecular engineering, and her lab develops sequencing analysis tools to study and fight cancer with an emphasis on alternative splicing. And so we're really excited today to hear about her work. And with that, please join me in welcoming Dr. Angela Brooks.
Oh, geez.
Yeah, I see it. Let me see. I can try to, let me see if I can try to turn off the laser pointer. That might help. Let me see if I can get that. Man.
Let's try how soft is big. And then can you exit up from there? No, it's still up there.
Or, turn off. Oh, that's okay. Oh,
Oh, here it is. Great. Okay, nice. And now we can
Okay.
swap displays, but I like it to show up.
there's a way to get it from here.
Oh, okay. Oh, nice.
That was great. Yay. Oh my gosh. Okay. Welcome. Thank you so much for the organizers for inviting me. It's really great to be back at UCSD and yeah, seeing people I've met before and haven't seen in a while, and looking forward to meeting new people today. So my background originally started in so my background, my PhD work was to look at how RNA splicing is regulated. And when I moved to my postdoc, I was interested in cancer genomics. And the month I started my lab, all these papers came out showing that splicing factors were significantly mutated in cancer. So I said, okay, well, I guess I'll stay in splicing and cancer. And so when I started my lab at UC Santa Cruz, I was really excited about what was then kind of newer long read sequencing technology. And so a lot of what my lab has established our projects in is of how we use this long read sequencing to better profile cancer. That'll be the topic of my talk. So cancer is a major cause of death, and it's a big reason why we study cancer. And a lot of what we're trying to understand is how do we better treat cancer. And so one of the, this is a very simplified view of the goals of the cancer genomics community is, well, let's just sequence a whole bunch of cancers and look for what are commonly mutated genes. So this is an example in, say, lung cancer, where if you take about 500 tumors, then you look at which genes are mutated. You see about 30% of cancers mutate the KRAS gene, about 11% get mutations in the EGFR gene. And then once we identify these patterns, then we can say, oh, these specific mutations would be really good targets, since they're very prevalent in a lot of different cancer types. And so one of the challenges, though, I think in this in the field is when we target specific mutations with drug, a big challenge is mechanisms of resistance. So a lot of patients overcome the treatment by, say, secondary mechanisms, some of them via splicing or other ways. The other challenge is that we've been kind of looking at cancer gene alterations. Kind of the way the analogy I like to think about is this elephant analogy. I don't know how many of you have heard about this, but if you have different people that are maybe can't see the elephant, but can only touch the elephant, they might be looking at different parts of the elephant. So say someone who's just touching the trunk of the elephant, they think, oh, this is a snake. Maybe someone that's touching the tail of the elephant, you know, feels what it feels like. It looks like, maybe it seems like it's a rope. And what we've typically done in the cancer genomics field is sort of like this, and we've been using short read sequencing technology. So we're sort of using short reads. Some people do DNA sequencing, some people do RNA sequencing, and then they ask, how is this tumor altered? So what are the somatic, so one group might look at somatic mutations and another group, okay, wants to look at splicing. But again, it's this idea that we're all trying to look at and understand what are the genes that are altered in cancer, but we're all looking at it at these different angles. And so the premise of the research in my lab is really, how do we harness newer technologies like long-read RNA sequencing, and can we actually see the entire elephant, look at all of these features all together with this one method and one type of analysis, and really see the complete picture of what the cancer genes are. And so to highlight why this might be interesting to look at, I'll just highlight a couple features that we might want to look at together. So somatic mutation and splicing. First, how does long-read sequencing help us look at splicing? This is a cartoon of that. And so in typical short read sequencing, you would have, say, poly A RNA. And sorry for people on Zoom, I just realized you can't see me here. So through poly A RNA sequencing, you would capture the transcripts, and then you would only sequence kind of subparts of that. So if you had two different isoforms that differ via alternative splicing, so I've kind of highlighted that here in the boxes, but you have certain regions that are shared by both, say, isoforms or versions of the gene. So when you do the cDNA sequencing, you might get a fragment that maps to that shared region between the two isoforms. And so you're not sure, did that read come from isoform A or B? And so for quantification, for isoform detection, this can be a real challenge. However, with long-read sequencing, you theoretically can sequence the RNA from start to end, and you would get exactly which full-length transcripts exist. In addition, what's really powerful is if you have somatic mutations or some kind of variants that are present, with long-read sequencing, you can actually see which isoform does that variant get expressed in. And here in this example, again, really simplified cartoon, but we may see these, what we're calling isoform-specific allele bias, where you might have a variant that only is present in the context of one isoform and not the other. And that might be important to understand what's the then functional consequences of these variants. And in most cancer genomic studies, when we look at variants, we're not asking, well, what isoform does that mutation fall in? And that might be important because if you're, say, trying to study the function of that mutation, what if you pick the wrong isoform to then functionally characterize? And so again, the premise of what my lab's working on is how do we really have this comprehensive view of all the different alterations, but using just long-read RNA sequencing. So in order to do this, we need bioinformatics tools to examine the long-read data. And so a lot of what my lab has been working on is a tool called FLAIR. And the first problem we tackled was to look at oh, sorry. So the first problem we tackled was to look at splicing. So how do we use long reads to examine splicing? So again, FLAIR, what it does is it takes long read data, and at the time when we developed this method, we were using a lot of Oxford nanopore technology in particular, which had a lot higher error rate. And so we wanted to account for that in trying to generate high confidence transcripts that weren't sort of misrepresented because of these errors. And so the way FLAIR basically works is it takes raw reads, it maps to the genome, and then you get this initial alignment. And then we incorporate either annotated splice junctions, or what's also very useful is other orthogonal, say, short read splice junctions to sort of correct the splice junction boundaries. Then we group the splice junctions together by similar junction patterns, and then we collapse them into a set of sort of non-redundant transcripts. And then we do this sort of second pass read alignment and read assignment to generate this high confident transcript reference. So our initial work with FLAIR was to look at changes in full-length isoforms associated with somatic mutations in splicing factors. So we worked on looking at alterations associated with SF3B1 and CLL, and also U2F1 mutation in lung cancer. We also developed FLAIR to look at direct RNA sequencing. So this is a cool method where you're actually sequencing the RNA molecule, so you have the potential to look at RNA modifications as well. We also looked at full-length transcripts associated with inflammation. So not cancer, but we collaborated with Susan Carpenter at UC Santa Cruz and actually found these really interesting novel isoforms in the innate immune system. And what's been really great, and for students here or trainees that are working on bioinformatics tools, when I was a graduate student, I wrote this tool called JunkBase. It's like really horrible to run and really hard for other people to run. And so when I started my lab, I really wanted to make FLAIR really easy to use. And so what's been great is that many researchers have been able to use FLAIR for their own research. And we're constantly still improving on this. If you have problems, let me know. But this is always a challenge. But FLAIR is not the only tool out there. So this is probably not comprehensive. I forgot to check this morning for new tools, but this is a non-exhaustive list of long-read RNA-seq tools that have been published over the past few years that perform the same kind of analysis. So take this long-read data, try to assemble transcripts. And the challenge then is, every method when they get published, they use their own benchmarks of how their method is better than other methods. And in addition, in the field of long-read RNA-seq, since it's so new, I think it's challenging for a newcomer to come in and say, okay, well, I want to do this analysis, so which tool should I use? And now you have this long list of tools and you don't know which one to use. Another big challenge for the long-read field has been the fact that there's these two sequencing platforms, so PacBio and Oxford Nanopore. And a lot of questions that I still get is, I want to do long read, but should I do Oxford Nanopore or PacBio? And a lot of data sets are using different platforms. So again, which one should we use? And so it was actually Ali Mortazavi, who's pictured here, who's part of the ENCODE project. And he kind of put together this initial just informal meeting in Barcelona with other people that were interested in long read technologies. And we kind of decided, we think the field is ready to maybe have like a more systematic community-based evaluation of long read methods. And so we formed this consortium called the Long Read RNA-seq Genome Annotation Assessment Project, called LRGASP. If people are familiar with previous, there's been CASP and EGASP and RGASP, so this is sort of the next iteration of that. And this work was published last summer. And this was a huge collaboration with ENCODE, GenCode, many other groups. I'll continue to acknowledge everyone. And so as part of this effort, what we did was we produced a large set of publicly available long read data sets. So we had a total of about 427 million long reads collectively from all of our samples. This was a big effort. Multiple labs contributed to the sequencing. So we did human, mouse, and manatee. So manatees, probably the FAQ most frequently asked question is why manatee? And so the reason is we wanted a non-model organism to evaluate transcript assembly when you didn't have a good genome. And one of our collaborators, Anna Canesa, was in Florida and had manatee collaborators. So that's why we picked manatee. And they're cute, so it's, it doesn't hurt. So we had these species, we had different sample types. We had single cell lines, we had cell mixtures, we had spike-ins. We also did multiple library protocols. We did cDNA library preps, direct RNA, R2C2, which is a method to get higher accurate cDNA libraries from Oxford Nanopore. And then we also did this method CapTrap to capture capped RNAs. And then we had 3 sequencing platforms. So what was great is these are the same RNA aliquots that were sent off to different labs to do the library preps and the sequencing. So it's a really unified data set that you can do really great comparisons with. And this is, still publicly available. So if people are interested, feel free to dig in. So we broke up LR-GASP into 3 challenges. The first one was transcript isoform detection. The second one was isoform quantification. And the third was de novo isoform detection without a good genome. And just for the purposes of time, I'm going to focus mostly on just describing the results from challenge 1. So one way that we tried to make this this effort a real community effort was sort of really engaging the research community at multiple steps. And so we had these. So I was part of the core organizers of this effort. But we also did a lot of outreach to the research community, in particular tool developers. So we produced the long read sequencing data, but the evaluation benchmarks and how we were going to evaluate was an open discussion. So this was the time of the pandemic, early times of the pandemic. So we would have these Zoom meetings that we just publicly announced on what was then Twitter, and people would join. And we would discuss what would be the right benchmarks for this. And once we created these benchmarks for how we were going to measure transcript detection, for example, then we even provided the evaluation scripts that were going to be used for evaluating the method. And so this was, I want to emphasize this because in previous efforts, like with these types of community benchmarking, in the past, there was sort of this moving goalpost where, people say, okay, submit your predictions, but then during the analysis, people would change what the benchmarks are and stuff. So we were trying to make that fair and say, okay, this is exactly how things are going to be evaluated. Here's even the script. So if you improve your method. Okay, so then with all of that in hand, then we had some, we provide the data. We said, okay, submit your predictions by this deadline. And then the tool developers then submitted, and also, sorry, a side note, we wanted the actual tool developers to submit as well, because as you know, you can run a tool, different parameters. And so we really wanted to make the fairest possible evaluation by having the tool developer themselves decide what parameters to use to run their tool. So they predicted the model. They provided their transcript models and transcript quantification. And then there was a subgroup, again, try to make it fair, a subgroup of the organizers that did not submit predictions. Those were the individuals that were involved in the actual evaluation and validation. Again, trying to make this as fair as possible. Okay, so that was the overall design of our LR-GASP effort. And again, we're trying to understand and evaluate transcript detection with long read sequencing. So there were 11 research groups that participated, including my lab. And what's shown in this graph is just the number of isoforms that were submitted from each lab and each tool, or sorry, each tool, and then also for each library type. And so what you can see from this is, again, this is the same sample of RNA that was sequenced, different libraries, and then the tools had to determine what isoforms were detected. And so you can see a lot of variability in the transcripts reported by each tool, where, at the lower level, Lyric, reports only maybe 5,000 transcripts or so, whereas another tool, ISO-IB, reports, hundreds of thousands. And so this was just one example of, why we felt as a community that this kind of effort was really necessary. Because if one is trying to do long read sequencing, like which approach should you use, which library prep and which computational approach, you can get many different or very different answers depending on what you choose. So here are the benchmarks we use to evaluate these methods. And the one I'll kind of touch upon a lot is these spike in RNA variants. So these are, this is a company that creates these synthetic RNAs. These are sequences that aren't thought to exist in the human or any real species. And you can spike it into your sample and then you can sequence them. They're polyadenylated. And so you should be able to pick it up in just your normal library prep. And at the time, we actually worked with Lexogen. They had these, what were called long serves that were previously not available, but they gave us early access, where they had these serve isoforms in the past, shown in the middle. But one challenge is that the length of those RNAs capped off at about 3,000 or a little less than 3,000. And so they added these long serves that actually ended up being really useful in some thinking about evaluation. So these long serves, the largest is something like 12,000 base pairs. So this is a really busy plot, but I'll try to step through it. So what this represents is the performance of sensitivity, precision, F1 score performance metrics for all these different tools, and all against these long serves, just those long serves. So what you notice again is a lot of variability. Again, warranting the need for such a benchmark. And I'll draw your attention to the left two columns. Those are Bamboo and Flare. And so I'll highlight this. So the long serves really highlight some of the reasons for these differences. So one is that certain library preps give better coverage of long transcripts. And the other is that certain pipelines are heavily weighting the annotation more than others. So you can see here Bamboo, which actually in the end performs really well in all metrics. But for the long serves in particular, it gets pretty much 100% accurate, 100% sensitive, 100% precision, it performs very well. Whereas Flare is much more variable, that depended on the library prep. So for example, what's in orange is the cDNA PacBio library. So Flare performed really well with those, but not as much for the, say, the cDNA ONT libraries. So again, this is just highlighting what I just said. Okay, so why is that? Well, when we sequence with all these different methods and all these different platforms, they all have different features of the data. The data have different features. So on the left plot is the read length distribution. And so you can see that the cDNA PacBio library, also maybe the R2C2 ONT libraries, tended to sequence much longer RNAs or cDNAs. The middle plot is sequence identity, so how accurate was the actual sequence. So we had all the PacBio libraries had the most accurate sequence. Second, kind of the R2C2 libraries were really good as well. And then the last was that the read number, so the read depth. So how many reads were sequenced for each library. And there's multiple dots because for a given sample, we had multiple replicates. So, for CapTrap, ONT, and cDNA ONT, we had a lot of reads per sample or per replicate that was sequenced, whereas much lower for the other libraries. So I'll focus in again on those, the PacBio cDNA libraries and the ONT cDNA libraries. And so the PacBio libraries had the longest read lengths, the highest sequence accuracy, but lowest sequencing depth. And so the performance on these long serves really highlights how certain library prep methods are better at sequencing long transcripts. So what you can see, so this is one of those long synthetic RNAs. Here is a coverage plot of that RNA using the cDNA ONT library method. And here is the coverage for the cDNA PacBio library. And so you can see for the ONT, there's a huge dip in the coverage, kind of in the middle of that transcript, whereas in the coverage in the cDNA PacBio library, just notice that the bottom is 300x. So you get at least 300x coverage or so across the entire transcript with that library prep. So this kind of explains why the different tools perform differently. So Bamboo, even though there isn't full coverage of that transcript and only partial transcript coverage, Bamboo says, okay, that's an annotated transcript. I don't need as much support for read evidence, so I'm going to call that a real transcript because it's in the annotation. Whereas FLAIR, the way we developed it, we didn't want to heavily weight the annotation. We wanted to make sure there was read evidence of that transcript existing. So we more heavily weight read evidence. And so that's why the FLAIR performance is much lower than the Bamboo performance for detecting that transcript. So then in contrast, when you have a library like the cDNA PacBio library, where the coverage, you get a lot of coverage, the read evidence is consistent with the annotation, then FLAIR performs just as well as Bamboo. And so I just want to highlight this because it's not necessarily one is better than the other, but it's just something that one should consider when they're trying to decide what tool to use. And maybe in different situations, depending on the library prep you have, you may want to make a decision about which tool you want to use. So for transcript identification, one of the things that was really surprising is that longer and more accurate sequences are preferable to having more reads. And why this was really kind of mind-bending was, I think, for those who study or have worked with RNA-seq, short-read RNA-seq, if you want to detect rare transcripts, you just sequence more, right? That is not the case with long reads. So, and I'll hopefully try to explain why. So here again is the read length differences between, again, the cDNA ONT and the cDNA PacBio. Sequence identity is very different, so definitely more accurate in the PacBio space. But the ONT libraries were sequenced much more deeply. So then when you ask, okay, you have these libraries, now what I'm showing in these plots is each dot is a report from a tool. So the reason there's many replicates is there's different tools sequencing or reporting the number of transcripts given a certain library. And so what I want to highlight again are those ONT cDNA libraries and the PacBio libraries. So what you can see is, so again, the left plot is still showing read length, but what you're seeing is that with the longer read lengths of PacBio, then you're actually detecting more transcripts than the cDNA ONT libraries. Also, sequence identity plays a role as well. A higher sequence accuracy, you're probably going to detect more transcripts as well. And then the number of reads. So this is where it was a little bit surprising that the number of reads didn't necessarily mean you got more transcripts detected. Again, very different than the models of how we think about short read. So here is a cartoon that hopefully, if this isn't making sense somehow, explains it. So you have this transcriptome. I'm representing this cartoon. You have some set of short RNAs, and then you have kind of a gradient short to long. So if you have a certain type of library prep, you're sequencing that limited kind of slice of the transcript length. So that's kind of represented by the width of this green box. So this is saying you have library A and you're only sequencing the transcripts that are about that length. And then what's shown in the height of that box is the read, say, kind of cartoon representation of the read depth. Now, if you have another library that maybe has a more dynamic range of the transcript lengths that it sequences, you can sequence more longer transcripts, even though your depth is shorter, the population of RNAs that you're sequencing is much broader. And so then it doesn't matter if you're sequencing more deeply in the first library, you're still only capturing that limited population of RNAs. So again, we think that sequencing longer transcripts actually will help you with transcript detection with long read sequencing. Okay, and then so from what I've kind of shown you, it makes you think, okay, wow, PacBio, maybe that's what I should be doing my sequencing with, because it so far has performed pretty well. However, I want to emphasize, and what was great is we had these different library prep methods in our LR-GASP effort. And so really it comes down to what cDNA library prep method are you using to put into your PacBio or ONT libraries. These sequencers, actually ONT has a longer sequencing length, but the limitation doesn't really seem to be the sequencer, it's the library prep. So here's one example from this study. So we actually had two different library preps done for the PacBio sequencing. So one was this CapTrap approach from Roderick Wigo's lab. And then the cDNA PacBio libraries were generated from Barbara Wold's lab. And so they had differences in the read length distribution. So the CapTrap PacBio libraries had a shorter read length distribution than the ones from the Wold lab. But they had pretty much identical sequence accuracy and read depth. And so, quick look at the performance of those libraries, we see again the better performance of the cDNA PacBio libraries shown in orange regardless of tool, because you're able to capture, say, the long transcripts. Okay, so that was one benchmark I kind of stepped through and some of the lessons learned about long read sequencing from our LR-GASP effort. And then one other benchmark I'll quickly show is that we also use orthogonal data to evaluate. So we asked, okay, from all these tools that are reporting transcripts, how many have support from short read RNA-seq? We also did cage-seq. So this is looking at the 5' cap of the RNAs, and then the 3' end, so that's quant-seq. And actually, there's a lot of variability in the way tools were developed. Some tools don't care about the transcript ends. And so we wanted to evaluate how much are these tools reporting the accurate ends. And so FLAIR ended up being one of the top performing tools on many benchmarks, one of them being this orthogonal support. And so this plot is showing on the y-axis the percent of novel transcripts reported by the tool that had orthogonal support. And then the x-axis is the percent of annotated transcripts provided by the tool that also had orthogonal support. So this also to say that annotations aren't always going to be correct for your given sample. And so this orthogonal support was our way to evaluate that. And so FLAIR is kind of at the kind of top right corner as it's trying to balance the accuracy of annotated versus novel transcript detection. So some key just takeaways from that effort. Each benchmark has its strengths and weaknesses. we really needed multiple benchmarks. I didn't go through all of the benchmarks just for time. And so there were many tools that did well on multiple benchmarks, some that didn't do well on multiple benchmarks. So fortunately, Flare was one that did well on multiple benchmarks. So we were really happy about that. And then one thing that was, I think, really surprising to think about for long read sequencing is how longer and more accurate sequences are preferable for transcript detection as opposed to just having more reads. And what I didn't have time to talk about is our quantification evaluation, but briefly, read depth is important when you want to quantify. So that's the kind of flip side. For detection, read depth might not be important, but quantification, it definitely is. And then if you have multiple read support, or if you have multiple methods that identified a transcript, then we had a higher validation rate. So I didn't go over as we actually had some side validation of these transcripts as well. Okay, so what I kind of talked about here is, you know, how we're using long read sequencing. I've kind of focused more on this sort of transcript detection. And now I'm going to layer in this variant incorporation. So how do we improve transcriptome analysis and incorporate variants? And so Flare, you know, again, I kind of stepped through how we started with splicing. And now we have a version Flare 2 that actually incorporates RNA editing. So I'll talk about some cool things we've found with long reads for RNA editing. So with Flare 2, this is actually the version of Flare that we use for the LR-GASP effort. So this was our improved version of our tool. So essentially it takes the read alignments, and it also looks, so how it improves a lot on, say, annotated transcripts is we check the reads directly against annotated transcripts and see which reads fully support annotated transcripts. And then we say, okay, that is a read for an annotated transcript. We don't need to look at that anymore. But then if something's not annotated, then we kind of go through our standard Flare pipeline, which I covered before, where we use some orthogonal support, correct splice sites and such. So then in the end, we'll get these high confidence isoforms that are both from annotated isoforms and potentially novel ones. And then in addition, we take those same reads and then we run a variant calling tool. So we're not doing variant calling ourselves. That's a whole nother area, but we've taken sort of standard variant callers for long read methods so that we can detect what variants are on those reads, and then we can integrate that together to get these variant aware transcripts. And the first type of variant that we were interested to look at were A to I edits. So A to I editing is a type of variant that actually only exists at the RNA level. You're not going to see this change at the DNA level. It's caused by ADAR, which is an enzyme that converts the adenosine to an inosine. And then that inosine looks like a mismatch in the RNA-seq. So it'll look like a G. So it's been known that A to I editing has been implicated in cancer. It's thought to alter splicing, can alter codon changes, change RNA structure, for example. And so we wanted to integrate this into our long read analysis. So this work was from Allison Tang, who also was a core developer of Flare. So she did a lot in her PhD. And we took these lung cancer cell lines and had control and knockdown of ADAR. And then we did, took the cells, did RNA extraction, and then both did Illumina sequencing and did this R2C2 method, which is gives you higher accurate cDNA sequencing from Oxford Nanopore. And I'm only going to show this one vignette from this study because I think it's pretty cool. So this was an example of a significantly downregulated A to I event that we detected with long read, but not short read. And that's because of hyperediting. So what I'm showing here is an IGV snapshot of individual reads that came from our control knockdown sample. And then down below is from our ADAR knockdown. So we can see in the ADAR knockdown in this sort of general region, a loss of a lot of these variants that you're seeing in the reads. And in particular, in this arrow, I'm highlighting kind of clusters of many different variants. And so this is the correct type of A to I change that we expect. And since it's lost in ADAR knockdown, we think these are bona fide A to I events. However, when you look at the short read data, matched short read data, you don't see this hyper editing in the control knockdown. What's happening, and actually if you look more closely, is there's a drop in short read coverage. And so because of the hyper editing, there's a lot of A to I changes that are happening in a clustered region. So when you're trying to map a short read to that region, you're not going to be able to map it. And so that's why we're seeing this drop in coverage. And so there are some tools to look at A to I changes in short read data. They're really specialized tools. But with this, it was easy to just look at the standard mapping tool and for long read data and be able to see these variants. So this is kind of another additional benefit of long read data is we can see these types of complex variation that we might have missed with short read. Okay, so now I've showed you some of our stuff with FLAIR2 where we looked at RNA editing and splicing. And so what we're working on now is to kind of get the whole elephant. And so we have a version called FLAIR3 that we're working on right now that incorporates fusions. It'll do fusion splicing. We can look at allele-specific expression and somatic mutation. And so why this is also interesting is then now that we have this complete picture of all the variants that are present in the long reads, then we can have better functional interpretation. So why might functional interpretation be more important? So one thing that's also been kind of overwhelming to deal with if anyone has looked at long read data themselves, there's actually a lot of isoforms that you see for a given gene locus. It depends on the gene, obviously, and the gene expression, but you can have gene loci that, maybe some of these are very rare isoforms, but if you collectively look at all that are detected, there's just a lot of them. And so, for us, we care about, what is the function of these different isoforms. If you're trying to look at each individual isoform and how they're differentially expressed, it might be kind of complicated. So what we're doing also in the way we're analyzing this data is we're grouping things together based on their predicted protein sequence. And also if it's predicted to be a target of nonsense mediated decay or degraded or non-functional, an unproductive protein. And so the way FLAIR3 works is one part. I don't have the fusion part up here just for simplicity since I'm not covering it in detail. But we take our FLAIR transcripts, our reads, we align the reads actually directly to the transcripts. We find that that's gives you better variant detection instead of aligning to the genome. And then we get a set of our variant aware transcripts. And then from the variant sequences of those transcripts, we're then looking at the predicted amino acid sequence. So we have, we're launching sequencing of primary lung cancers, both tumor normal. This is a project with Alice Berger at the Fred Hutch Cancer Research Center. And we've done a small pilot so far. We have just 2 samples. We've done PacBio IsoSeq, and we have a patient that was, we have a male and female patient, different stages of lung cancer, different smoking statuses, and we've sequenced matched tumor normal. So I'm just showing one example from, and this is really preliminary, unpublished right now. So this is a known variant, BRAF V600E. It's a known driver mutation. And we're seeing this isoform specific allele expression. So that this is that this somatic mutation is only being expressed in a specific isoform of BRAF. So it's a little busy here, so I'm gonna try to step through it. So if you look at isoform usage, so just not thinking about variants, we have about 5 or so, yeah, 5 different variants that are expressed at all at different levels, right? So the first 2 columns is one normal tumor pair, and the next 2 columns is the second normal tumor pair. And so what you're seeing, what you can kind of see from just this first plot is that you see this upregulation or kind of isoform bias that's observed in both samples for the tumor. So they're specifically upregulating this BRAF 220 isoform. That's the way it's annotated in Ensemble. When you look at then the variant that's expressed, the V600E, so we can see that it's expressed in the actual reads. And then we look at the predicted sequence. What we're seeing is that, again, that variant is only present in the isoform, this 220 isoform context, not in the other context. And then when we look at now predict the amino acid sequence based on all the variants, we can see, it kind of pops out like that this is a very tumor specific transcript, and that it would be predicted to have this V600E. Okay, so that's kind of the little flavor of what we're doing with FLAIR-3. And for time, I'm not going through all the different ways we're looking into improving FLAIR-3. But really, this is kind of our goal is we're trying to take long-read RNA sequencing and have the capability to look at all these different features of a cancer gene to really have a better understanding of what genes are mutated in cancer, how they're mutated, and hopefully for better functional interpretation. And so with that, I'll think, a big part of the talk was about this LRGAS effort. And so specific mention to Anna Canesa and Francisco Pardo Palacios, who are the leads of the real evaluation for the transcript detection. And then finally, my lab, so some key people, Allison Tang developed initially FLAIR and FLAIR-2. Cameron helped a lot with initial FLAIR. Colette Felton has developed FLAIR-3. And then Taylor Wan and Namrita have been helpful with the lung cancer project, as well as Alice Berger, her lab. And so I can take questions now.
Hi. Well, wonderful talk. One question I have, though, is that when you're benchmarking these different tools, do you take into account algorithmic efficiency, runtime efficiency, memory time efficiency? Because just from my personal experience, I was working with like ONT cDNA data, like really large libraries, and some of those tools, they might have changed them, but they were like they would have taken, some would run less than in a few hours, others would have taken a month, we estimated. And I suspect it's probably because they were not built, they were probably built off of like some of those PacBio or dRNAs. So yeah, I guess that's my just simple question. How do you evaluate algorithmic efficiency for these different tools?
Great question. We decided not to evaluate that mainly because we actually thought about this, of making all the tool developers create Docker containers that they would have to upload so that we could test, right, what their efficiency was. But then we thought that was going to be way too much of a burden, honestly, like it was already a burden to have them submit data, then to have them submit like their package in a certain way. We were worried about that. So that's one reason why we didn't go that route. I should say, yes, that is a big issue. I say for our lab, I actually just hired a part-time software engineer to help with that for Flare, because that we definitely, as we're, the sequencing depth's increasing, we're interested in single cell. There's like, a lot of large scale efforts now, and a lot of people are collecting data, and that's a big issue. So yeah, we're working on it. I don't know about other tools.
Thank you very much.
Yeah.
So, well, thank you for a great talk. So my question is about the therapeutic angle of that, and really, really fascinating what you said at the end, that the isoform distribution or a specific isoform of that distribution is responsible for V600E, for example. And so now instead of developing a drug which is specific to that mutant, you can actually maybe control the distribution. My question is, do you know how to control the isoform distribution and what's responsible for that?
Yeah. So there's some approaches we're doing in this collaboration with Alice Berger. So there's some CRISPR-based methods we're planning to, if you knock down a specific, say, only exon, for example, how do you control, and then look at the functional changes or how that affects whatever function. So we are looking, what I didn't talk about, we are interested in drug response as well. And so for certain genes and say the RAS pathway or targetable pathways, we're interested to know how are these isoform alleles correlated with drug response. So CRISPR methods, overexpression studies as well, not ideal, obviously, but those are the approaches that we're taking to try to tease out isoform specific function.
How's it going? Thank you so much for the talk. I was interested in the slide that showed the number of transcripts identified for each tool.
Mhm.
And I was wondering what caused kind of the large variation where some tools were predicting under 50,000 transcripts and others were predicting 330,000. Is it that some tools are like missing transcripts and others are over predicting kind of these new variants, or yeah, what causes that variation?
Yeah, I don't know all of the reasons. I know that it kind of, I guess conceptually, I think it comes down to how you collapse things. So each individual read, at least the way Flare works is, we see an individual read and we say, okay, that is the same thing as this other read. So we're going to collapse that into one, right? So that's one layer. If you're not correcting for errors, potentially, then you're going to have a lot of heterogeneity. And so you're going to have all these unique transcripts present. So then you're going to have, instead of 1, 2, or 3, or 4. So I think that's a big part of it. We didn't systematically dig into all the reasons, but that would be just from my knowledge, general knowledge, that would be one. And then the low end would be just the filtering. Like some people decide different cutoffs of how many reads support an isoform before they move it forward as a high confident isoform, because that can depend on different tools as well.
Thank you.
Hey, Angela, that was really nice. So I guess you kind of alluded to my question already, but I was wondering what you think the potential is for doing these types of analyses at single cell level. So looking both at detection and quantification.
Yeah. So we're also for this work with Alice Berger, we're planning to do for some subset single cell sequencing. So I can say there's actually a recent paper that came out from Genentech. Ashley Byrne is the first author, where they were doing single cell capture. And that's actually the approach we're going to take as well. So, so genes we care about, we design capture probes for those, and then only sequence those through this through single cell, because a lot of the issues is, we care about certain genes, and then you get the dropout issue. There's a lot of noise as well in the single cell library preps. You get a lot of internal priming, for example. So this paper talks a lot about these artifacts in single cell. So the capture methods really help to clean that up. So you get the most out of the reads you get. And then one thing, I was talking to Hannah about this too, that kind of, I haven't thoroughly like examined this through all the data sets, but one surprising thing for some preliminary long read PacBio data we've looked at with 10x. One thing to realize is that these libraries, the way they optimize these library preps for, say, single cell is to try to capture as much RNA. But in that sense, you're actually biasing to shorter transcripts. So if you care about a long gene in your single cell analysis, you really want to be careful because you might not actually capture it at all. And actually, anecdotally, I talked to a researcher recently and they were having this issue as well. So that's why I haven't like systematically looked into it in real data, but just anecdotally through some of our own looking at sequencing. I think even for single cell short read RNA-seq, the cDNA, the underlying cDNAs that you're sequencing from short read RNA-seq are going to be shorter genes or shorter transcripts. And so this is something that, I want to talk to more people about, because I think if you care about long reads or longer genes, or, we might want to think about different types of library preps. And this is something like we're even for bulk, we want to test out kind of better, longer RT methods and stuff to get longer transcripts. Yeah.
Thank you.
Mm-hmm. Yeah.
What are your thoughts about
the communication and use of this data in pathways? That for decades, we've represented things as gross oversimplifications of the world. We knew they were gross oversimplifications, but we didn't have the data. So now you're generating this data.
No. Oh my gosh. I'm gonna try to pull up a bonus slide. I think we're low on time. We'll see if I can do this quickly. Let's see if I do this quickly. Okay, I'm gonna do, this is from. Okay, that's working. Yeah. So this is great questions, something that we're really excited about looking into more. So KRAS, for those who might know about, KRAS is one of the most frequently mutated genes, and especially in lung cancer. And there's two different isoforms of KRAS, 4A and 4B, that are annotated. Most people kind of focus on KRAS 4B because it is the most highly expressed isoform of KRAS. But there's this other isoform of KRAS 4A that's more lowly expressed. This is like a distribution in tumor normal. But what you can see in the 4A case, that is the isoform that's upregulated in tumor compared to normal. And this is based on short read data. But what we're also seeing this in long read data. So I'll just, so in this lung cancer pilot we did, we can sequence these full length isoforms. And so we're seeing also from between normal tumor, it's the 4A isoform that's being upregulated. Now, there are papers that show that 4A has a different affinity to other effector proteins in the pathway, but it hasn't been characterized as much. So this definitely, KRAS, if you look at, there's tons of pathway diagrams with KRAS and like what it interacts with next and how it fits in the pathway. But how much there's a difference in 4A versus 4B and what does the pathway look like with different isoforms is unclear. That's definitely, we're interested in, in that question as well. And maybe the, cancer, the tumors are going to tell us like what, it obviously is selecting more for the 4A for some reason. So why, I don't know. But Yeah.
Oh, one more question, anyone? All right, with that, let's give Angela another round of applause. Thanks so much. I think we have some drinks left behind. If people want to grab something on their way out. Maybe a few stray slices of pizza there.
It's okay. So we have a gift. So thank you. Enjoyed your talk. Yeah, thank you so much for coming. And I think